We're gonna go ahead and start our seminar for today, and David Sullivan is going to introduce our speakers.
Thanks. I'm going to introduce Dr. Dan Hanley, neurologist who has had intersecting trails with Peter Agre and other people throughout Johns Hopkins for decades. We have a busy agenda with, I think the main theme is how a clinical trial engine can shed light on biology and equipoise. But I'm going to go ahead and introduce Dan, and he'll walk through a schedule and the other speakers. Thanks, Dan.
Thank you, David. It's wonderful to have this opportunity to talk at the School of Public Health and in the MMI. You're a very important set of people who generate ideas, and we're kind of at the other end of generating the ideas, designing the trial and collecting the evidence. And we wanted to talk about two instances of doing that are MMI instances. And we're going to have regular clinical presentations, clinical scientific, and then a presentation of how the trial operation works to support the biologic idea. So you can see David Sullivan is going to be paired with Karen Lane, who supported his COVID convalescent plasma trial, and Shmuel Shoham is going to be paired with Nicole McBee. And then we'll have time for a discussion afterwards. These are our panelists and their relationship to everybody else. There are no conflicts. We want to do three things. Tell you about resources that are available to support clinical trials. Define some examples of clinical trial operations that are supported and that were key to the execution of the main biologic hypothesis. And built into this is a very important story of trials and pandemic operations, which David and Karen are going to tell us. BIOS supported both David in his trial with Shmuel and Arturo and Kelly Gibo, and is now helping Shmuel and Kelly and David with an RSV trial. Our basis is this tree of trials that we did, starting with brain intensive care, which is where I started, but we've spread out to a lot of other areas, and you can see some of the things we do, which we're going to talk about. We do three main things: trial planning, grant organization, which is the argumentation that something's important, and then trial operations. And we're going to talk particularly about trial operations today. These are our speakers, and without further ado, David's going to tell us about the outpatient COVID-19 trial.
Thanks, Dan. I'm able to share sort of results start from finish. The main point is high antibody level plasma is effective at reducing hospitalizations. The same plasma did not prevent infections, but it also was able to neutralize future variants. A brief starter case: a 70-year young woman with diabetes, BMI of 27, presented in July with cough for a couple of days, diarrhea, dizziness upon standing. At that time, there was no outpatient interventions, and the trial was a 50% chance of something that may have worked. She enrolled. And then the next week, she progressed and had to go to the hospital with the typical ground glass opacities in the back of the lungs, rather than in the front. This is why we put people on their stomach. We found out later that she was randomized to control, and she actually received convalescent plasma while she was in the hospital and did well. So at the start of the well, it took 2 years for drugs to arrive. The start of a pandemic, everyone was naive, and you have passive and active immunity. And on the spectrum of passive is convalescent plasma, which was transfused in January 2020 in China. So it was available almost right away. These other preparations take about 6 to 8 months to be ready. This is the time to phase 3 efficacy trials, inpatient and also outpatient. I mean, checking the dates, we were among the first for outpatient antibody trials with the monoclonals about a week later for the first patient first visit, and vaccines even later. Then on the bottom is just the time to emergency allowance, so lower evidence of proof, and then the vaccines on the top. So what is the randomized clinical trial? It's a randomized control trial, I'm sorry. In this case, we took high levels of antibodies and a double-blind patient and study team, randomize a large group to achieve power, and we monitor infection or hospitalization. In this case, for early treatment, a 50% reduction in hospitalization, but not in plasma. But it's a simple design, but it's hard to implement. And actually, Arturo, March 18th, emailed me and said, David, I need a clinician to help write some grants. The next day, I was looking for advice, and Dan Hanley and BIOS were one of the first ones to offer real help with the study startup. And so in rapid time, just a matter of weeks, we had already identified through their clinical trial network possible sites. Here we had a bunch of cold calls with more than 24 teams, finally selected 13, and then made the first patient first visit. We also had rapid collaboration, cooperation on the regulatory sideline. Everyone was rushing to hospital, and I said, well, my mother's not going to want to go out and join society if we don't have outpatient measures. So we drafted an outpatient RCT and submitted it in about 2 weeks. Approval in 2 days by the FDA, and about 2 weeks with the IRB, including pregnant women because of safety data from the hospitalization. And we had through the BIOS network, we're able to set up sites, 23 places in 2 waves. This is sort of the pace of our recruitment or transfusions associated with the pre-alpha, alpha, and delta period, another 20. And then our punchline was that it was efficacious in reducing hospitalizations robustly. It wasn't a fragile trial. It was safe. And this was enough that a week after public announcement, we were able to, the FDA issued an emergency use allowance. So this was really in 2022. They also issued approval of emergency for drugs at that same time. The past 2 years, we've been really working on building an FDA package. And so we also answered a lot of biological questions. I'll go into that. But then December 10th, FDA did finally get approval for a biological license application to make it available for the immunosuppressed, although they commented that it was sufficient evidence that it worked in those that were immunocompetent. Some of the biological questions along the way, an important one, working with Andy and Sabra and Lee, was that if you take the upper deciles of plasma, yeah, there's a pointer, that we have good, effective, this is plasma collected pre-alpha that neutralized Delta variants and also Omicron, but you can see a lot of the low-level antibody plasma did not. Arturo published with others that hospitalized patients responded better to locally acquired plasma, but it's probably with a medium titer direct match work better in neutralization. But if you have high titers, then you don't have to have an exact match to the circulating variants. And this was an important point that was followed up with some other studies. Coming from a drug background on malaria, I was really interested in dose and time, early and with high dose. And I worked with collaborators across the street, that's Arroyo, on figuring out that we only gave about 2 milligrams as opposed to 200 to 2 grams of monoclonal antibodies. But that was enough when given early, and above 2 milligrams, we had 0 hospitalizations. If we had more numbers, it would probably, the more accurate counts, around 20% hospitalizations of the total hospitalizations. But again, it gets down to this trial did reinforce the biology that early antiviral action with antibodies is the method. Then this is working with Mario Catarugli across the street. I reached out to him early in March too about, well, we need an antibody test that's going to be important going forward, March of 2020. And he had diligently followed 15,000 people, mainly to show that the plasma that we used was low titer, still worked, was efficacious, but after vaccination and going up, he had about 50 to 100-fold increase in the antibody levels. And this is just the road to the FDA and the approval. Well, the clinical trial engine shed light on a lot of biological plausibility. We had trials on different phases of the disease: infection prevention, early treatment in pediatrics, and also those hospitalized, newly mechanically ventilated. At Hopkins, it wasn't powered to show an effect. In Belgium, it was recently showed that a high titer plasma does work in that case. Sabra had a very seminal paper on age, sex, and hospitalizations, increased antibody levels. Arturo and Scott McConnell showed that antibodies uniquely can degrade spike protein that was important, and we looked at future variants. There were a lot of studies on secondary outcomes of other clinical studies. Kelly and others were involved in guidance and perspectives, and Shmuel. I really want to highlight the nurses. They were the backbone. I learned a lot being in the clinic, and was also, well, it distracted me from the world around me. I could focus on this trial, lots to do. This was a person in the trial, Dr. Sullivan team. Thank you for are truly healthcare heroes who gave us hope. And a long list of names of people at the trial sites. So we all stood on the shoulder of giants, stood on the shoulders of BIOS, who helped provide the framework, weekly meetings, communication to the DOD. And I do want to thank the funders, especially the DOD, Bloomberg for a $3 million, and the state of Maryland for $1 million to get us started back in April of 2020. I'm going to turn it over to Karen, who's going to sort of go through some of the more of the nuts and bolts of how to get a trial started. Thanks.
Thank you, David. Hi, everyone. So it's been a privilege working with Dr. Sullivan, Shoham, and Jibo, and all of the COVID convalescent plasma teams during the acute COVID stage. Our clinical trial team and coordinating center started 25 years ago with our own research program. And now we are helping others across the country to build their clinical trials and programs but it's always nicest when we can work with our colleagues here at Hopkins. But whether we work across the United States or here at home, no matter how many times we travel it and with whom, the clinical trial road is filled with roadblocks driving us to what we do, which is translational team science and innovating to improve how we conduct the clinical trial. So our job is to maintain and manage consistency and compliance for all the project components, starting with the research strategy to finally sharing results. Knowing from this experience and what roadblocks and risks are likely to happen at each stage of the trial, this is what enables us to skillfully help others strategically for creating consortia. How do you build platforms, training, recruitment, and retention, and in the end, communicating results in publications and also sharing with our valued trial participants. Throughout the clinical trial life cycle, our overarching goal is not only to conduct trials more efficiently and cost effectively, but also as a laboratory for improving the clinical trial process. And for the COVID trials, we used tools and we embedded many of these tools, including an accelerated startup, managing the IP inventory, and a marketing program. When we first teamed up in March of 2020, an important focus was guiding the new teams quickly through a startup. And we demonstrated in the COVID trials that the time for startup could be reduced from hundreds of days to as little as 90 days. And you can see that with the COVID plasma trials, the time was even faster than that. Time was so important then, but time is important for all trials. A fast startup can really provide an additional 10 to 15% time for more recruitment when the time comes. These trials taught us that we could do it, and now we've done it in more than 12 trials. The pandemic drove us to breakneck speed in trial design and organization. From concept in March, we submitted two grants, 2 INDs, 2 IRB submissions, and enrolled our first patients in June. We surveyed 65 CTSA sites across the US and activated 26 sites while still finding our funding. And then we worked with transfusion medicine to rapidly set up a plasma coordinating center in time for those first patients in June. The fast pace made it difficult to maintain consistency and compliance, but we did. Our communication plan was complex as this organizational chart. What you see here represents the combined knowledge and expertise of multiple PIs, multiple cores, multiple coordinating centers, overseeing project components across the consortium. We acted immediately and adjusted as we went along. Everyone committed to weekly updates, scientifically and operationally. We had every site investigator at every site on a call every Thursday afternoon. We met with our funders and regulatory officers every week and met daily with all of our cores. This beautiful flow diagram was created by Dr. Sullivan to show the story of our inventory system and how we managed the collection of 3,000 units to ultimately the transfusion of 1,351 units at 26 locations. Even though it depicts the flow of study research plasma, it also represents the same flow across all the components. The flow was the same for the serologic testing of antibody tests. It was the same for supply chain management and specimens, for our data platforms, for our QA monitoring, and for the rapid communication of all the protocol updates. It is the perfect representation, as David said a moment ago, of all the aspects of a randomized clinical trial engine, the complexity of a well-run trial, and not only quickly, but with an eye on FDA compliance for submission. On the roadblock side, the pandemic virus and strategies to accelerate pandemic countermeasures were constantly evolving and forcing adaptation. Often, site elements were in place only to have clinical spaces change, nurses leave, coordinators and staff pulled back to their other jobs and clinical duties. Then the monoclonal treatments arrived under the FDA emergency use allowance, and then vaccines were approved. All of this required the coordinating support of the BIOS staff and 5 regulatory bodies monitoring 5 performance scores and 84 site teams. Recruitment challenges included politicized COVID-19 information and competition for enrollment. And when we did talk to patients, only 1 to 5% that we contacted were interested in the study. This forced us to adapt an accelerated and hyper-focused trial recruitment pathway. We launched both English and Spanish websites and engaged a HIPAA-compliant call center. And with all of these marketing strategies, we were successful. We went from 200 randomizations to more than 800 randomizations in 6 months. In 2023, the early treatment trial was awarded one of the top 10 clinical research achievement awards in the country. And another extraordinary fact is that we had 100% compliance of our primary endpoint. There are now more than 60 publications and more impactful science still evolving. And as Dr. Sullivan mentioned, there are new international guidelines based on our trial results. Much of our work is supported by the NIH Trial Innovation Network. That's who we are. As a trial innovation center, we link investigators to more than 60 CTSA hubs, matching trial opportunities to best suited local investigators at each of the 60 sites. And perhaps our tools and our network can be of help for your next multi-center clinical trial or your trial someday. We are packaging our tools and training to create open access and REDCap platforms and website toolboxes so that investigators and project managers like yourselves can use what we use with us or independently to improve your metrics. And for those of you who are leading a trial, or if you're a trial site, we have materials you can use even though you are not directly involved. We have turnkey, shelf-ready, free tools you might find helpful for your trial or your trial to be. We are here to help you. We encourage you to pursue our resources and our excellent consultation teams. We're here, so thank you. And with that, now Dr. Shom will talk about our new work together.
All right, next slide. All right, everybody, Shmuel Shoham. I'm going to talk about how to go from molecule to market when there are limited financial resources. I'm going to talk about a trial of RSV immune plasma as treatment for RSV upper respiratory tract infections in high-risk immunocompromised patients. And this is a product that is not attractive to large pharmaceutical companies. It's not going to make anybody billions of dollars, but it will, in my opinion, save lives. Next slide. So the concept is that immunocompromised patients are at highest risk for severe or even fatal RSV infections. Patients with hematological malignancy, solid organ transplant, and assorted other immune suppressive medications can lead to a high risk of progressing from RSV, which typically causes fairly mild infections, to progression to pneumonia, respiratory failure, and even death. RSV infection is very common. There's RSV season, which essentially is the same as flu season, and it confers short-lived immunity. That's why you can get infected again and again and again. And RSV passive immunoprophylaxis has been used, but not treatment. So RSV immunoglobulin is used in people at risk for developing severe infections, including young children, and then monoclonal antibodies, palivizumab and nirsevimab, have also been used. They work, but they're unavailable as treatment for adults. Next slide. There are 3 currently available vaccines for RSV currently, one made by GSK, one made by Pfizer, one made by Moderna, and all of them will lead individuals that receive that to have high neutralizing antibody levels. And of course, we know that selected patient groups may not get access to the vaccines, and then those that are immunocompromised may not be able to mount the kind of vaccine response that would be very protective for them. Next slide. And here's some work that was done here at Hopkins by my colleagues, Dr. Caraba and others, showing that transplant recipients do have a response to vaccination with one of these RSV vaccines, but it's neither uniform nor very robust in some patients. Next slide. So passive immunotherapy, as you've learned from Dr. Sullivan, has been effective as treatment for COVID-19, and we were curious to see whether we can take that to the next stage, which would be treatment for RSV in high-risk patients. Next slide. Currently, there are available passive immunotherapy options, but they're inadequate. There is IV, that's intravenous immunoglobulin, IVIG, and there's particularly RSV IVIG, which is IVIG that has high levels of anti-RSV antibodies, but it's not currently approved for treatment of RSV. It's approved for treatment of patients with hypogammaglobulinemia in general. It's very costly. It's not easily scalable. And in getting on the phone to colleagues around the country, essentially no one has used it because it's so expensive. Other lessons that we learned are that respiratory viral infections in general, and I think this applies to RSV as well, you got to treat early and you got to treat with high antibody level preparations. And that if a patient has low respiratory tract infection and they don't progress after, say, a week of symptoms, it's unlikely that they'll progress. So we want to grab patients early when they're at highest risk, but we also don't want to grab patients that are going to get better regardless of what we do. So we have to be selective in the patients that we are choosing for treatment. Next slide. And when all this stuff was floating around in our minds, one, we had an idea of a product that could make a difference in people's lives. Two is we needed to find the right patient population that would benefit from it. And three, we needed to have funding to do a clinical trial proving or disproving that a high-risk patient population it can receive this therapy and have improvements. So we turned to the Trial Innovation Network, and thankfully they agreed to take us on for consultation. And by we, I mean me, Kelly Gibo, and Evan Block. And it's been very helpful. Next slide. And some of the things that we work with them is one is talking about what the product would be, and using their experience in terms of starting to work toward an investigational new device application. And so the product would be high titer post-vaccination RSV immune plasma. So as I mentioned, people that get vaccinated, particularly people that are not immunocompromised, get high neutralizing antibodies. And the combination of them having had previous RSV, as all of us have had, and on top of that, getting the vaccination creates a situation where we can have high titer RSV immune plasma available for use as treatment. Next slide. Then we came up with a hypothesis, which is that RSV immune plasma is safe and it could be associated with a reduced incidence of progression from upper tract disease to lower tract disease. And then working with the Trial Innovation Network, we came up with some study aims, and that's to show that RSV immune plasma is effective and it's safe, and then also to study the pharmacokinetics because we have Dr. Sullivan's expertise in the area. Next slide. This is how the trial would be set up. We would find 200 high-risk patients with RSV, and I'll tell you about what a high-risk patient is in a minute. And then 100 of them would get usual care, and 100 of them would get usual care plus high-titer RSV immune plasma, and we would see how they did in terms of the outcome. Next slide. And the outcome of importance is development of lower respiratory tract infection. Can we prevent people with upper respiratory tract infection from progressing to that very potentially deadly and challenging lower tract disease? Inclusion criteria are, this is a study for adults with RSV upper tract disease that has been present for less than 5 days, and an immunodeficiency scoring index of 6 or above. Next slide. So the immunodeficiency scoring index, the goal of that is to identify those patients that have a high chance of not just getting better over the RSV by themselves, as most people will do. So this particular group of patients that, and they get scores, as you see, for each of the different parameters. When the score exceeds 6, their chance of progressing to lower respiratory tract disease is about 50%. And about a third of them will die. So we want to grab those patients, and then maybe patients that are just a little tad bit less sick than those. So a score of 6 or higher is what we're going for in this study, so that we can try to prevent people from progressing to lower tract disease. Next slide. And then after they receive the treatment or the placebo, in addition to usual care, then if they do develop lower respiratory tract disease, then we want to grade, was it lower respiratory tract disease with no oxygen needed, or was it all the way to, God forbid, death? Next slide. The reason we came up with 200 patients wasn't because 100 is a nice round number. I mean, there was a little bit of that, but generally it's, if we assume that with that ISI score of 6, we can identify patients that 35% of them will go on to progress to lower tract disease, and assuming that the product will have a 50% reduction in that, then having a power of 80%, we see 97 patients in each arm. There's a question as to, does usual care include antibody therapy? Usual care can include anything that people do. It can include ribavirin, which some people feel is as good as placebo. It can include IVIG. It can include any kind of other therapies that people might think will help. So at this stage, we don't think that there's any good therapy, and we're not sure that IVIG makes a difference in treatment, but the which is pooled immunoglobulin, but the convalescent immune plasma has high neutralizing antibodies. So we think that's where going to be the high octane stuff. Next slide. So we approached and by we, I mean the Trial Innovation Network approached a range of hospitals that are known to have these kinds of patients, and quite a bit of them showed interest, and a substantial number of them also gave us some numbers, and those numbers show the number of heart and lung transplants, if we choose to include those, and bone marrow and leukemia patients, which we are definitely targeting, that have RSV in a given season, and it's 541. And if we assume that about 10% of those are going to meet the ISI 6 or above, we can assume about 50 patients per season over a season would be eligible. And my experience has been that we usually get about 50% of the people that are eligible will agree or meet criteria to be in the study. So we can assume about 25 patients per year in the US. Next slide. However, we have decided to go beyond the US and include Brazil. And the reason for Brazil is that RSV seasonality in the US and is flu season in the US, which is November to March, April, May. It is the inverse of that in the southern hemisphere. So by having Brazil, we can go year round in terms of recruitment. So the 25 or so patients that we think will be enrolled per year increases to something higher than that. And then that allows us over a 5-6 year period to enroll enough patients to be able to make a definitive statement as to whether high titer RSV immune plasma can be effective in reducing the progression from upper to lower tract disease in these high risk patients. And by reducing them from going from upper to lower, we can hopefully save lives. Next slide. You've heard a lot already about safety data management and clinical monitoring, so I won't repeat that except to give you the highlights in that Dr. Henley and his group know what they're doing. They have a real track record of taking a concept to clinical trial, and then from there getting it to the point where potentially it can go to market. And you've heard from Dr. Sullivan how that was done with COVID convalescent plasma, and we hope that we'll have a chance to do that as well with RSV immune plasma. Next slide. The significance of this I've touched on in various parts of this presentation, but this will be the first to determine the safety and efficacy of this product. It could offer a novel treatment for RSV, something that's desperately needed in immunocompromised patients. There's a growing population of patients that will need therapy for RSV, and we hope that this could be that therapy. It's a low-cost therapy and that it can be scaled rapidly, and it can help inform the regulatory approval. And then one other thing is having an original idea is extremely important, and that was COVID convalescent plasma. Continuing that to the next product allows us to go from a one-hit wonder to something that is more consistent and has a real future. So we're hoping that by extending the concept of COVID convalescent plasma to RSV immune plasma, we will set this idea on a stronger footing. Next slide. All right, the next speaker will be Nicole McBee. She's going to talk about team science and creating a successful trial strategy.
Thank you, Dr. Shoham, and thank you all presenters and for everyone for being here. So I'm hoping to, in this last presentation, to make some connections. So you've heard a lot of information in the last 40 minutes. So looking at team science and how we help investigators such as Dr. Sullivan and Shoham and Jibo and Dr. Block, others, bring that thought and turn it into a strategy and a project as part of a program. So you've heard about BIOS Clinical Trials Coordinating Center, and celebrating our 25th year. Can't say it enough, we're pretty darn proud. And we are a full-service academic research organization. We are a division within the Department of Neurology over in the School of Medicine. And we work with investigators within Hopkins, the entire Hopkins, so School of Public Health obviously included, as well as investigators outside of Hopkins. So we're willing to talk to just about anyone. And you can see on the right there all of the various resources and services that we provide to help projects such as CSSC and RSV come to fruition and funding and success. So our center, our coordinating center, adds value through partnership. And you saw some of those examples in Karen's presentation, where we look at consortium management, so finding the sites, credentialing the sites, activating the sites, all of the things to do to find the patients to collect the data. And through that, we also have a 60-day study planning program where we take those first 2 months of funding and nose to the grindstone, develop the protocol and the MOP, the manual of procedures, and all of the different things that you'll need once those sites are activated and the trial is running to support that lovely Sankey diagram that Karen showed and all of the many, many moving parts of a multi-center trial. 90-day site activation, as Karen showed, it's a data-driven process and it is built on many, many years, not just the 12 that we've successfully done it in, but it's something we started as a concept 25 years ago in one of our first multi-center trials that we've now honed and validated and is available for resource for investigators such as yourselves. Quality management system is across the board from start to finish, starting with a risk assessment all the way through your study closeout, making sure that patient safety and trial integrity, data integrity are at the forefront of everything that we do as a coordinating center. And then, of course, the key opinion leaders. We can't forget the clinical trial expertise, the subject matter experts that we bring together as part of our program, our core, for your benefit to essentially give you the input and the questions and the thought-provoking discussion to help you as the scientists bring the biological science and turn that into a strategy that will be feasible and realistic and successful, built upon our clinical trial infrastructure, operational infrastructure. So as Karen showed, we are forefront BIOS Clinical Trials Coordinating Center, but we are also a trial innovation center within the Trial Innovation Network. So you are here. And in that way, we're able to help investigators at the 60+ clinical and translational science awardees. These are 60+ academic institutions and their affiliates across the US. And we do that through consultation so that we can help any investigator who's looking for input and support for the development and conduct of multi-center clinical trials and to benefit from all of the stuff we know. And that's the whole message here. We want to disseminate what we know. We want to help others do what we've learned how to do successfully. And the TIN, the Trial Innovation Network, over the last 9 years now, we're in our second funding cycle of funding. The first fund was 7 years. We're in our second award of 5 years. We've done 463 total proposals across those 9 years, and taken those to either through initial consultation or we've provided with resources, discrete resources such as recruitment feasibility assessment, recruitment and retention planning, expression of interest, electronic health record-based tools and resources. What David and Schmoltz showed you, how we are able to reach out to those CTSA consortium, other sites to say, are you interested in being a trial site to enroll patients? Do you have the patient population? And it's a data-driven phenotypic query into the EHR record as opposed to a subjective, yeah, we have those patients. We also look at community engagement studio so that we're making sure the investigator has refined inclusion-exclusion criteria, as well as study design, consent materials, marketing materials for recruitment. We partner with the Hopkins JHM School of Medicine IRB as the single IRB, and we've been doing that for the last 9 years since the common rule changed, and we help educate investigators on how to work with them, as well as support those processes, and then standard agreements. So the reason the Accelerated Startup Unit works, the program works, as Karen showed, is because we have worked very hard to find every efficiency we can squeeze out of the SIRB and standard agreements pathway, as those two things are usually what take the longest starting up sites. We also help locally. So we're integrated here at the Hopkins CTSA, otherwise known as the Institute for Clinical and Translational Research, or the ICTR. We talk in alphabet soup all day long. And we intersect with the Hopkins ICTR or CTSA in the same consultation mechanism via a trial advisor program. And that allows us to work here locally and then funnel to nationally those projects like RSV and CSSC that are ready for that national multi-center strategic component. Through the TAP, we really, our goal, as usual, through the TIN as well, is is to have an outcome of your consultation that meets the needs of the investigator. And that can be various things. So one potential outcome is referral to the TIN. But we are organized as partners with the ICTR and all of the many, many resources that are available to Hopkins researchers through that mechanism, that funding, our clinical trial operations, and then we also work with the Center for Clinical Trial Evidence Synthesis, their consulting service. We're partnered with them as well to have as robust as we can a support system for consultation. And that expert consultative support is for single and multi-site translational clinical trials. Essentially, if you have an idea, you're ready to start enrolling human patients, you've gone from bench to bedside or first in human, or you've done a pilot and you're ready to go multi-center, that's when you come talk to us. And we're there to give strategic advice around the trial design, around trial startup, enrollment, data quality, good clinical practice, IRB, human subjects research, leadership and management of research teams. We're going to help you know what you don't know, start asking questions to get you thinking, to help design. And all of this, we collect data on everything. So use of performance metrics, not only to show you what we do and why it works, but also then to compare your project and make sure it's on task to be successful. Comprehensive safety and risk management, we're with you along the way to make sure, once again, human safety and trial integrity are of utmost importance. These are the team science approach. So connections are our things. We like to connect investigators through all of the various resources here at Hopkins and nationally and through BIOS. And then our use case, to give a little more information about the RSV plasma consult, which is an active consultation right now. You're seeing hot off the press information that Dr. Shoham shared. The consultation came in to the Trial Innovation Network and was assigned to BIOS as a Trial Innovation Center in April. We had our introductory call, we had our kickoff call, we worked it through our workflow process for consultation. We are working with Dr. Block and Dr. Shoham and the Office of Clinical Trials here at Hopkins so that they can submit an investigator-initiated IND, investigational new drug application, to do this project under FDA management. And then as part of that initial consultation, we decided this will be a collaborative effort. We will move on to a comprehensive consultation to where we will continue to design and flesh out the operational logistics for feasibility to then get a funded application, which is down here, targeting May, but that remains to be seen. So lots of work along the way. We're 5 months in, 6 months in, 7 months in, but the consultation service is here to review, and it's based on the investigator needs. So if you have a concept and we're able to give feedback and then you're able to do what you need to do, it doesn't have to be a complex relationship, but those that need it are able to use our infrastructure. So for the consult, as of right now, just in those 6-7 months, we've been able to help the investigators refine their design and to refine their intervention from a 2-part effect size of high titer RSV and then treatment to really focusing on that treatment intervention. We've expanded from national US only 20 sites to now including Brazil for the reasons that Dr. Shoham talked about for feasibility and making sure we can recruit year round. We did expand from a 5-year project to a 7-year project, and that has been through conversations with our potential funder, NIAID, and then sample size looking at power and the primary outcome that has evolved through the consultation. We went from a starting point of 60 patients to 200 patients, as Dr. Shoham described. So the consult value add is really to take home that it is so fundamental that as you're preparing your idea, your concept, and your funding strategy, your program officer at wherever you're submitting for funding, that's your friend. Talk to them. It is absolutely fundamental to make sure that they're a part of it, there's interest, and their guidance is so valuable. Our collaboration through the IND, as we did for CSSC, and that we will do for RSV to support the investigators as well. And then consortium identification through TIN expression of interest and the site assessment survey instrument that Karen talked about as well to help credential those sites, again, data driven. just to bring it home, I'm running out of time. You can see here through the consultations, the TIN, the national, and the TAP, the local, are quite diverse in their clinical domains. So the investigator brings that biological science, BIOS as a TIC, or through the TAP brings that clinical trial expertise. And through those things, we do good work. It doesn't matter to us what the biology is. We rely on the investigator and our subject matter experts to help us guide and refine clinical trial operations that fit the biology. And then satisfaction-wise, we do collect satisfaction results through the TAP and the TIN. I'm showing TAP here. We do have a 60% response rate, which is pretty high. I'll be proud of it. For satisfaction surveys, it's usually around the 20% that people respond, and it's been overwhelmingly positive. So on a 10-point scale, most 93%, 97% responses have been in the 8 to 10 range. With neutrals, there have been no negative comments. How can you access us? You can go to the ICTR website, or you can just Google ICTR TAP Trial Advisor Program. The website link is here. There is a maker request. It will be routed right over to us. We will talk to everyone. So if you're not even sure, go ahead and submit it. We'll talk to you. And if we're not the right people to talk to, we'll connect you with who we think would be able to help you. That's all I have. Thank you.
Thank you, Nicole. So we're going to have a little panel discussion. And for those of you who survived the panel discussion, there are some refreshments out in the lobby afterwards. Arturo, do you want to say something?
What's striking about what happened was the individuals who do not know each other, you know, basically came together in the very least to create a structure that delivered. And it's a remarkable example of what I consider what is known as Tocquevillean situation. So Tocqueville visited the United States in 1931. He noted that the United States, as compared to Europe, whenever something had to be done in a city, that the citizens came together and this, whereas in Europe, by on central authority. And here's an example. Here is an example that something emerged. I think it's one of the strengths of our society that this required no direction. What it required was people willing to come together and put together an idea, and you see a lot of the participants sitting up front. And we are into rough times and a lot is happening, but I think that we need to also take advantage of our strengths, and it is a strength of American society.
Thank you. So questions for all of our speakers. Yes.
So I'm wondering what your take beyond what was done for sale to be, I'm wondering about your capability to work with other centers.
So capability to work in non-US centers. Karen, do you want to speak to that for a second?
Certainly. And yes, we've been quite international in our experience. We participated and led clinical trials in most of the countries in Europe, Asia, China, Australia, Israel. We've been Brazil, we've been there. So we have experience throughout.
Essentially, you find like-minded groups and you partner a little bit in the way that Arturo mentioned.
Correct. And for countries we haven't necessarily had direct access in or direct experience in, part of what we do and the reason we have been able to do this for 25 years is we rinse and repeat. So we take what we know, we use that to guide us into the new area, and then we make sure we talk to other like-minded individuals to find people that will help us be successful. So countries such as Africa, for example, where a lot of infectious disease research is done, we have people we can talk to essentially for subject matter experts to guide us.
Yeah, and Lindsay might be able to talk to, you have some experience.
Yeah, in my previous life, I used to do HIV research. We collaborated with the School of Public Health. They were looking at phylogenetic analysis of the HIV virus and how it was spreading throughout Africa. So yes, we have a lot of contacts and actual experience in my own country.
And Kelly might also talk about capabilities of BIOS and Hopkins in general to set things up overseas.
I think we were just gonna say, one of the things we can do is leverage our colleagues who have contacts overseas. So one of the things we did with Brazil is we all reached out to our colleagues in Brazil and said, which of these sites could potentially work? We internally vetted them and our friends, already. And I think was helpful in sort of thinking through which sites would have been potential options for us.
The electronic phenotype helps a lot these days too. So you go to a colleague and then you get data. Other questions? I have one. Finance and compliance are really important. Lisa, why don't you tell us a little bit about the back office of finance and compliance and minute by minute oversight of large trials?
Well, it's very, very important to make sure that you have enough funding to be able to conduct whatever study you're working on. Clinical trials, when you're involved in clinical trials, there's a lot of things that come to mind. Flexibility, negotiations, prowess, and the capability to understand the needs of the trial. So communication is vital. This group is an expert at developing a communication system very depthly and are able to really communicate their needs. And so my job as the financial manager of this trial was to make sure that we had adequate money to make things very swift in nature in terms of signing on clinical sites. There was a lot of contractual needs, a lot of contractual things that needed to be identified and worked on rather quickly. We were able to work with our counterparts in Jura and ORA to establish those mechanisms very quickly. Of course, compliance is the utmost consideration. And sometimes compliance can be a real roadblock. But we were able to very quickly and be able to assign a sense of urgency with all of the compliance that we needed to comply with. Amy Gawad really helped a tremendous amount with the IRB applications in terms of compliance. She was a wonderful colleague to have at all times, just to be able to talk things over. She's a doer, that's for sure. So it's really important, that the term like-minded people is rampant in this conversation. And that's the thing that I've come to learn about BIOS is that they're ready to rock and roll. They have systems already in place. Really very, very wonderful people to work with.
That was a paid advertisement. But Amy is a local resource that no one should underestimate. Sponsors, you brought up the word sponsors, and you were the key pivotal point in communication with sponsors. But it starts even earlier, and I'd like for David and Kelly to comment on communication with sponsors. Maybe David in terms of COVID-19 and Kelly, the ongoing communication with NIAID for RSV.
Sure. Well, I mean, starts with Arturo, an idea in February in Wall Street Journal, and Fauci had commented on it later, and then turning immediate therapy does take money. And I was distracted on hydroxychloroquine. I had started off with connecting Evan Block and Sanjay Desai on convalescent plasma about March 3rd. got distracted with hydroxychloroquine, and Arturo sent me a note. And really, it's with any idea, it's a one pager. The specific aim page that we know, but it's a one pager, and that's a vehicle for communication. But it also follows up with discussion and with sponsors. We had a DoD that was interested in our weekly meetings and was very participatory. There were some regulatory guidelines. There was the big final clinical study report, FDA IEC3 compliant. And so but then even, well, the communication with sponsors goes on after the trial, keeping them up with, I mean, they gave their hard-earned money with further results, FDA approval, other milestones. But even during the trial, there's still, well, with Bloomberg, it was almost monthly updates. Lisa had a big, strong hand in that. I also too. So, and the final thing on the budget is a strategy. It's not set in stone, and it's dynamic. But part of running a trial is well, paying attention to the sponsors and their program needs and how it fits in, and being responsive and flexible and adapting to that. Over.
Kelly, why don't you tell us about the first part of sponsorship and advocacy with RSV?
I think David's points are very important about the idea of engaging with your potential project officers early on and asking for their ideas and feedback on your aims. And one of the ways that you could potentially leverage your idea to what their long-term goals are. And I think we have worked with that with NIAID in terms of, we went with one idea where we had potentially a 2-stage proposal, and they came back and said, we really think this might be better as a 1-stage proposal, and we've had to revise and accommodate that. And I think that that's been helpful for both of us, so that we put together something that is both of interest to them and financially what they potentially think is a feasible project. The other thing I just want to say is that, as David mentioned, you come and go in with the best ideas and best intentions, but as you know, we saw with COVID and the rollout of monoclonal antibodies and the rollout of vaccines and needs for transportation, we had to adjust our budget accordingly. And so we would have a weekly meeting with the DoD, and David did a phenomenal job every single week of updating what are the newest things in the study, but also what are the newest things in COVID. And that was something I think was very helpful to our sponsors, for them to realize that we were paying attention to what these new developments were, and we were adjusting accordingly. And I think that information allowed for us to make real-time decisions or real-time budget allocations that needed to happen over the course of the trial that we couldn't possibly have anticipated in the spring when it actually launched.
And those adjustments and statements allowed for the 26 sites to continue to have equipoise because the relevance of the hypothesis that we were studying was maintained. Yes.
I'm curious about the breadth of your operations in terms of topics. Like, do you go to devices? Do you do non-infectious? Like, how far out?
We do devices. We have probably 8 FDA device approvals that we've helped with trials. The last one came in last Friday. We do drugs. We do small startups. We've partnered with bigger pharma. We go from fetal interventions all the way to Alzheimer's. We have some cancer experience, but cancer is pretty segmented. We're currently looking at 2 cancer projects, and a third one someone called in yesterday. So it's pretty broad, and you saw Nicole's pinwheel. There's a lot of colors there.
I just want to clarify, we don't do drugs, we do support drug trials.
Okay, but like, there might be people.
Well, I do drugs. They have me on drugs.
Mosquito release, modified genetic changes to insects.
Yeah,
Is that something you have experience with?
no, but we have a malaria expert,
Yes, you
Sullivan.
do.
Yeah, so we like complex interventions that are not a single intervention that can be proprietary. And we think that that's good space in the university for people who want to make management strategies and systems of care work.
I mean, I'll add to that. I think Dan Hanley and BIOS could tackle something as complex as the mosquitoes, and it's a strategy. It's the depth of, it's the experience on adapting and being strategic. I think with Nicole and Karen and Dan, you have a lot of collective wisdom on managing complex.
Other questions? I have one other that I want to go to. It has helped in both of these instances to have a written protocol, a set of sites that say they have the patients and are willing to do it, and some secondary electronic validation that there's some form of feasibility. But the other thing that once you get to that stage that helped tremendously in the COVID CSSC 004 protocol was a third kind of partnership or sponsorship, and that's with the FDA. Many people are afraid of the FDA, but having written documentation of what you're going to do and what your safety steps are lets you engage with them. And we engage with them on multiple levels. Two people in particular, Peter Marks and Janet Woodcock, were particularly helpful. Janet Woodcock asked to meet with David and Shmuel and Kelly and wanted to know about the trial plans and said the trial was too small, but she went out and got another $20 million for us, which really launched us from 13 sites to 26. And she argued for it. And it's just based around the facts. Peter Marks consistently looked at our filings. He was working an Operation Warp Speed, but decided he could help the pandemic better by going back to the FDA. And he consistently has looked at the overall issue of antibodies. He's worked with Arturo on strategies, but he worked very nicely. His division worked very nicely with us without any heavy-handedness in providing David with the pathway to eventually achieve a biologic license approval. And maybe I end the story a little bit with, there was a lot of skepticism about this approach. We were not warmly embraced by many of the strategic leaders, despite the fact that between 1890 and 1930, there were 6 Nobel Prizes on the role of antibody in infection. And it's nice that now 130 years later, after the first Nobel Prize, there's actually something that never existed before, a biologic license agreement to use serum as a treatment. And it's an accomplishment that everybody shares in, and I think is worth. There are refreshments outside to the right, and those of you who can join, we'd love to talk to you. Thank you very much.